Welcome Life Science learners to another installment of Life Sciences. Trust that you guys are well and excited about our lesson today. In our lesson, we're going to be focusing on the revision of genetics, genetic engineering, and a very important skill, writing essays. So I trust that you guys are excited and looking forward to our lesson. Let's get into genetic engineering, let's unpack some terms, and let's look at how we apply these terms in the context of a few questions. It's important that we recognize that this section is based on concepts that are derived on terminology. So it's important that you write these terms, guys, and have a list of these terms that you quickly refer to when revising or preparing for a section on this test. So let's move on to some core concepts that we're going to look at. We're going to look at the overview process of genetic engineering, and as I mentioned, we're going to unpack some concepts called revision on genetic engineering. So guys, it's important that when we look at this, the aspects of the section on genetic engineering are important. This is essentially a breakdown of the concepts that you need to have when looking at genetics and genetic engineering. We need to know all the terms around genetic terminology, and these include the terms alleles, locus, homozygous, heterozygous, dominant, recessive. Remember those. We've got to know patterns of inheritance, and these patterns are complete dominance, incomplete dominance, as well as codominance. The pattern in which characteristics which are inherited on the gonosomes we refer to as the inheritance of sex-linked characteristics. So we've got to know how to determine gender, which is in the inheritance of sex, but also the concept of how characteristics are passed down when they're associated with the X chromosome. So we refer to that as the X-linked characteristics or the sex-linked characteristics. The next aspect would be the concept of monohybrid and dihybrid crosses. It's important that you know how to lay out a genetic diagram that shows you a monohybrid cross, as well as being able to interpret the Punnett square in a dihybrid cross. The concept of mutations such as sickle cell anemia, hemophilia, color blindness, and other abnormalities, and understand how these are inherited by using images or charts called pedigree charts. We need to know the concepts of genetic engineering and the different processes that occur, examples of which are cloning, recombinant DNA technology, somatic cell nuclear transfer, and even a new concept called CRISPR Cas9, and so that's a gene editing tool. It's important that you understand that genetic counseling relies on information that you assess from pedigree charts as well as from genetic screening. The idea of paternity testing using DNA fingerprinting and blood groups is another aspect that you would be required to know when we're revising this section. So let's look at some terms related specifically to genetic engineering. Guys, the concept of genetic engineering is being able to engineer, change, make an organism that is genetically different, and that is either to the benefit of the organism or to the benefit of the person that is manipulating them. In this case, it would be the genetic engineer. The concept of using living organisms, biotechnology, using living organisms to produce useful products, and that comes from using bacteria, fungi, and viruses to produce the likes of vaccines, in baking, as well as using bacteria to produce things like human insulin or growth hormones. We got to know what genetically modified organisms are and the concept of GMOs, the concept of polyploidy and how organisms have multiple sets of chromosomes as a result of genetic engineering processes. The idea of using genes to treat certain conditions we refer to as gene therapy. And finally, the concept of cloning and how that is used in the context of creating asexually genetically identical individuals. The next concept which we use in cloning is called somatic cell nuclear transfer. It's essentially transferring the nucleus from a somatic cell into another cell. The technique of recombinant technology essentially points to how we recombine genes from one species into another, and that process creates genetically modified individuals. The concept of what a plasmid is as a vector, and how that is used to transfer genetic information. And what happens at the end of recombinant DNA technology is we have a recombinant DNA. There are enzymes that are involved in the process, such as ligase and a host of restriction enzymes. And these are tools that are used to cut the DNA and to stick them together. The next concept, transgenic technology, essentially transferring genes into another species or individual, and that creates organisms that are transgenic. The idea of selective breeding or artificial selection is where an animal breeder chooses individuals with specific characteristics and crosses them. And that's also seen as genetic engineering at a very high level, where we're selecting characteristics, also referred to as artificial selection. The concept of using vectors to transfer genetic information, and how individuals that are created show hybrid vigor, generally are having more greater variation or characteristics that are useful for a specific purpose. And finally, as I mentioned, the concept of CRISPR Cas9, which is essentially a very advanced and new technology in using of gene editing. So that's basically the ability to be able to use a combination of enzymes to edit out certain mutations or to change the genetic makeup of an individual. So as we have done these terms, let's kind of practice this with a set of descriptions and establish these terms. So the first question or description is a way of treating a disease by inserting new genetic material into a patient's cell. And that process is called gene therapy. So when we use individuals' genetic makeup and transfer that into an individual to treat a patient, we call that a therapy using genes. The next description, males and females with desired characteristics are chosen for interbreeding. Again, guys, that is selection of individuals, and we often refer to that as artificial selection or selective breeding, where we're choosing individuals with characteristics and crossing these. The next description, DNA formed by combining a segment of DNA from one source into the DNA of a vector. So that concept is combining genetic information into a vector is called recombinant DNA technology. Enzymes used to glue DNA pieces together, again, that is an enzyme which we refer to as ligase, and it's used basically to connect a recombinant DNA into a plasmid. Mating of organisms not so closely related, again, this encourages what we refer to as hybrid vigor, and that is referred to as outbreeding, where we try and bring in variation by crossing individuals that are not closely related, and that is called outbreeding. An organism with a foreign gene, and that's essentially an individual that we refer to as a genetically modified organism, or in some individuals could refer to it as a transgenic organism. So these individuals have got genetic information transferred into them. The altering of the genome by a technological process, and that is called gene editing or genetic modification. And finally, the use of plants, animals, microbes to create useful products is the use of living organisms, and that is biotechnology. So the process of using living organisms to create useful products is called biotechnology. Guys, that's a wrap for this segment. We've looked at some terms, we've looked at some important processes, we've tried to attempt a few descriptions and find these out. I trust that you guys have enjoyed this segment. Have a little comfort break, stretch a bit, get some oxygen into those muscles, and I'll see you in a bit.